righty, I think we'll go ahead and get started with our next Ivan workshop on the power of 2D and 3D NMR. At this time, I will pass it over to David Rice.
Hello, everyone. I see we have a very good group, and welcome back after what's been a sort of a summer vacation. Hope everyone has had a good summer vacation. So as Eric said, our leader today is John Ralph from University of Wisconsin, who's going to be talking about 2D and 3D structure determination. But before we get going here, let me hand things over to John.
Oh, thank you, Dave. And as usual, good morning, good afternoon, good evening, wherever you may be. And welcome back to the Ivan meetings. Dave mentioned we've had a nice extended Ivan vacation that we've all returned from, and back to work as usual. And back with us as always, Dave, Dan, Krish, Eric Schatzline, and the turnout is great. I guess you guys missed us. So we're back for some more autumn fun at this point. But yeah, we've got very good speaker today. Looking forward to John's subject material. And our sponsors, as always, MR Resources, Q1 Instruments, of course, our co-sponsor and sister company. Please do have a look at our sponsors. And but anyways, Eric, could you give us a little bit of an update on some future meetings that we've got planned?
Yes, I'd like to start with, we're still looking for presenters going into 2026. So if anybody has anything that they'd like to present themselves or know of anybody else that might like to do a presentation for us, that would be great. Our next two upcoming meetings are Tuesday, October 28th. We have a presentation on a quantum-inspired radio frequency for digital health by members of the Department of Physics at Federal University of Technology in Nigeria. And then on Thursday, November 20th, we have real-time C13 detection and the use of dual receivers to track polyl and I'm lost on that one, but it's Genevieve Seabrook from the Princess Margaret Cancer Center. So back to Dave, I guess.
Okay, great. I was reminded that October 28th is the anniversary of the founding of Ivan, and I've forgotten how many years ago.
I think 2016, Dave?
2016, yes.
That's right.
So
Yes.
not 9 years coming up. Yeah, you're right. We certainly can't overlook that.
Okay, well, let me hand things over to John Ralph from the University of Wisconsin, who will talk about 2D and 3D NMR.
So, first of all, thanks. Second of all, I feel a bit insecure in this group. Our group is very much NMR users. We're not NMR developers. The next two presentations make me feel even worse. The plan, though, is to highlight some of the incredible and unmatched value of NMR for dealing with a bizarre polymer that you might not have come across, and frankly, horrible samples. And basically, just to go over the way we like to make and authenticate assignments. That's probably very familiar to you, but we hope might put a fresh face on the power of and utility of NMR, something you already appreciate, but with some fresh examples. So, right off the bat, I don't want to rabbit on about our favorite molecule because we're supposed to be talking about NMR, but I think many of you probably have no idea what lignin is, so just a very quick introduction. Lignin is the stuff in plant cell walls that holds the cellulose fibers together. Obviously it's the world's most important polymer. Okay, maybe it's not, but it's the polymer that holds the fibers together in the plant cell walls and it allows trees to grow to these magnificent heights. But it's also the polymer that we need to remove if we want to get at the cellulose for paper or textiles. Also, one of the largest sustainable source of aromatics on the planet, and it's terribly underutilized. But and 20 gigatons are produced a year. This is quite strange for something you may know little about and may have never heard of. Right, that's about as far as we'll go with that, except to note some strange things about the structure. So it's produced from polymerization of two primary monolignals, these hydroxycinnamyl alcohols. They just differ in the degree of methoxylation. But unlike most other biological processes, this is purely chemical. Once you make the monomers, you send them to the wall, they make phenolic radicals, and they couple in a combinatorial way that's purely dictated by chemistry, and this has some interesting ramifications. So the main reaction is not these things reacting with themselves, but reacting with the growing polymer that can be represented like this. And I'll sometimes talk about the G and the S units, the G's with the 1-methoxyl and the S's with 2. I'll try and not get that too complex. But the problem is, like I say, it's combinatorial radical coupling, and so all you can do is assess the types of structures you get in the lignin, and I won't really show you where those are from, but you can assess the polymer based on the prominent structures in the lignin, and these are these beta O4, beta 5, and beta beta that I'll start calling A, B, and C, and hope to try and be a bit consistent with that, but not get too hung up in it. The big deal is that, again, it's combinatorial, but it's worse. Some of you have noticed that these units have 2 optical centers in each unit. The product is racemic, so it's stereochemically complex as heck. There's no defined sequence. Obviously, any single molecule has a sequence, but the polymer itself, the mixture, you'll never find 2 molecules the same. Unbelievable complexity. I'll just illustrate that for a second with 1D NMR. If you make a little dimer, it doesn't have quite the right side chain on it. It's a model for a dimer, but you can very easily see this beta ether has 2 optical centers. There's 4 optical isomers, 2 real isomers, so-called threo and erythro, and I've labeled them in green and red for these 2 protons, basically in a proton NMR spectrum. All you have to do is make a trimer, and suddenly you have 2 to the 4, 16 optical isomers, 8 real. We were very clever and managed to make components with one of these fixed as 1 isomer, the erythro isomer, and the other one S. So we've got 2 mixtures of 4 isomers. The normal 8 products is a superposition of these 2 things, and I think it's not hard to realize that if we broaden the peaks a little bit in a polymer and didn't enhance it this way, that you would see very, very little detail from these. They would be broad messes. And in fact, the lignin spectrum looks something like this, the sharp peaks not being anything real anyway. Very, very broad and horrible spectra. Great. Just compare that with these are only a couple of thousand molecular weight, by the way. Compare that with a 24 kilodalton protein where you can assign all kinds of peaks and you see a lot of fine structure. So we got a terrible mess to start with. Just back up again for a second. We've been lucky to collaborate with an enormous number of groups, all working to understand this polymer and improve its utilization. We've been lucky enough to get on covers and all kinds of things. I'm not going to talk about any of that, but I really do want to highlight the collaborators that we like to think that we do some primary work as well, of course, but that have usually contacted us because of our NMR, let's call it expertise, that we can help them define what they've done to the polymer, either by genetic engineering or with processing or whatever. All right, and probably many more than these. Okay, let's talk about NMR for this lignin, but actually also for whole biomass. It turns out that all you need to do to get remarkably useful spectra is to finely divide wood, for example, or the whole plant cell wall material. We like to extract out the extractable components first and just look at the cell wall, because that's what we're interested in. But you make these samples that then can be swelled in DMSO-D6 or slightly better in 4:1 DMSO-pyridine. This is probably the first time that the plant journal has ever put 2 clear tubes on the cover of their journal. And I won't go into how nice the spectra of the whole cell wall look, but we'll do some examples in a second. Yeah, what's this idiot trying to show Dr. Franklin Orr of the DOE? Many of you run NMR facilities, and if someone comes to you with an NMR tube that they turn upside down and doesn't flow anywhere, I think I can imagine what you would tell them to do with that sample. Uh, we've been told that ourselves. Turns out you get remarkably good solution state spectra despite that horrible mess. First thing I want to do, the first thing we have trouble with doing is explaining what the heck these C13 1H correlation experiments are all about. We've got very clever collaborators that really don't understand NMR, and when they look at these spectra, what they see is something like a Rorschach test, and it's worth trying to figure out how to best explain that. And the way I'm going to do this is with a simple little mixture of three compounds that I'll show you in a second. But basically, these HSQC spectra, as you well know, tell you more or less nothing other than that there's a proton at 4.7 parts per million attached to a carbon at 86 parts per million. It's useful in other ways because if you have overlapping protons, they're pulled apart by their different carbons. And if you have overlapping carbons, they're often pulled apart by their different protons. So the dispersion or resolution in these spectra is remarkable. But what do you do with them? Well, first of all, again, you know better than I do, these super simplistically come from applying whole sequences in which you separately put the proton and carbon nuclear spins through certain spin gymnastics. And with carefully selected time delays, you allow them to talk to each other and exchange information, resulting in this beautiful, for example, CH correlation experiment. That's not the one, by the way. There are many thousands of these experiments, some of which are incredibly useful. One that's underutilized is this HSQC-TOXY experiment. And the first thing that happens when you throw this up is people say, okay, great. All I see is there's more peaks now. What the heck am I supposed to do with that? How is this useful? It starts to become useful. I'm going to dim it down and show you what the compounds are. These are a mixture of the three primary dimers you get when you take that monolignol, the coniferyl alcohol, and you do the radical coupling on it. Sally, my wife, has a really cool way of making this lovely mixture of these three components very, very easily. But how does that help? What is this thing? Well, you start to realize that the spectrum on the right has sets of correlated or redundant, if you like, peaks. And it's because this experiment allows the protons to talk to each other and then to relay the information of everything in that coupling network down to their carbons. So basically each unit has 3 carbons and 4 protons. And so each proton sees each of the 3 carbons. Okay, each of the 3 carbons sees the whole 4 protons in that coupling network. So you realize these things all belong to the same structure, one of these structures. And I hope this works, this little animation. You can map these across then onto your HSQC, and you realize you've just defined these 4 peaks as belonging to the same structure in that mix. You can keep doing that. And that structure very quickly. If you're a chemist, you can figure out that it's this one, the C structure. And so we've basically defined that in the mixture. You can keep doing that. These peaks obviously relate to each other. You slide those across. You've just identified these green peaks. That's this phenylcoumaran beta 5 coupled unit. You do the same thing again. You've got these things, these little blue things. You raise that across and you've just defined those. This is this beta ether, but there's 2 isomers of this thing, so the other isomer is also there. You slide that across and you just realize that you have assigned everything in that HSQC experiment with the help of the beautiful redundancy sort of in the correlations in this experiment. Incredibly useful for assigning those peaks in a mixture like this. But the mixture is even more interesting or important. I'm going to show you how that relates to the lignin polymer itself. And to do this, I'm actually going to use methylated and acetylated dimers. So it's the same little 3-dimer mix, but we've methylated the phenols and we've acetylated it. And why would you do that? Well, most of the units in lignin are esterified in this phenolic position, so the methylation helps bottle that a little bit better. And acetates, if you make acetylated lignins or acetylated cell walls, these things are slightly better solubility and have slightly superior spectra. So what does the lignin spectrum look like? Oh God, it's awful. It's horrible. But perhaps you can see that it bears some similarity to this dimer mix. And in fact, if we slide that across again, do the same old trick, and zoom in a bit, you can see that actually you can define or you can assign those peaks rather unambiguously from that little model mix. The C component is obviously there. These green component, these B things, obviously there, and these beta ethers are obviously there. Even the two isomers are nicely separated. You can see that there's different environments. This is a complex polymer, so there's obviously another set here and elsewhere. So it's way more complex, way more broad because of that stereochemical complexity. But those assignments are very easy to make from that simple dimer mixture. Yeah. If you actually zoom in a bit further, you can actually even see, I can see this trio and erythro isomers of this beta ether unit. And I think you can see there's another trio set right here that obviously is because something different is attached either here or here. All right. Oh, a little sideline. Sometimes there's decades of mysteries that are in our research environments. This is one. These beta 1 couple units where the side chain's gone have been implicated in lignins for a long time. They're found in degradation products as simple molecules. But when you look by NMR, and we make model compounds again, and we know how to look for them, when you look by NMR, you can't find the dang things. They are not there. Just cutting to... Oh, and one other thing. For some reason, this phenol is always free phenolic. This one gets esterified. This one doesn't. What the heck's that about? So this was a mystery for decades. Finally, with the group in Sweden, Li-Ming Zhang and Yellowstad's group, we got beautiful spectra of a component that hadn't been assigned before. And basically with that HSQC toxi experiment, we related these peaks into one unit. We related these blue highlighted peaks into another unit. And you can imagine we can do this HMBC experiment where this carbon relates to all the protons here and to two of the protons here. So you know they both belong in the same structure. So it turns out that this beta 1 structure, before you degrade it, this is what it looks like in the native polymer. And that was not understood for decades and decades. Note that this thing is not a phenol. This chain can't extend for this N because you can't make a phenolic radical and you can't do phenolic coupling. So this is why this N never ends up being esterified. All right. Another little aside. Again, you all know this, so I feel a bit embarrassed, but NMR is diagnostic as all heck. Let me give an example of that. A long time ago, it was realized that there were parachimerates on grass lignins, these things. And where are they attached? The reason we want to know is because where they're attached tells you how they got there. So where are they? Are they on this gamma position or are they on this alpha position? And who cares? Well, again, it tells you how they got there. It was a beautiful study way, way back by Higuchi's group in Japan, but with UV. They made the two model compounds for these things, and they looked at the grass lignin and tried to determine where it was, and basically decided that it was about 60-40 alpha and gamma. Again, I don't want to denigrate that work. It was just fantastic at that time. But what does NMR do for us in terms of clarity for this? Where is this parachimerate on lignin? Well, you run the 2D NMR spectrum, and we know where alpha acylation would occur. It's right here. There's nothing above the noise level. Here's all the gamma acylated units showing up like a sawtooth. So there's no question unambiguously that there is no alpha acylation. It's all gamma. Everything is on this gamma position. And that, tells you something about how these things were created. One way is to take the monolignal and this parachimerate, presumably in its activated form, and join them together and make this monolignal conjugate that you then lignify. This piece lignifies the same way as it always did, and for reasons that I won't go into, this bit stays hanging around in the air, but always at this gamma position on the polymer. The implication of that is that this has to be a gene or an enzyme that stuck those two things together. And so, in other words, what we're saying is we've got these coniferyl and synapyl alcohol produced in the pathway. We've got paracamaraweal CoA also in the pathway. And so, presumably, there's an enzyme that takes those two components and hooks them together to make coniferyl and synapyl paracamarate. And when you lignify that, that's what's responsible for these gamma esters in the polymer. Usually we don't like these stacked plots for 2D, but I think it's quite useful in this case because it just literally shows there's nothing above the noise level in the area where there would be alpha paracamarates. Okay. Yeah, just an aside again. Once you've got a gene or an enzyme, or once you've implicated one, you look in a nice little model grass. Richard Sibou has well, first of all, in the wild type, you can see in the aromatic region peaks for paracamarate. You have a mutant that is deficient in that enzyme. Again, Richard Sibou, there's nothing there. You can downregulate that gene and you get a significant reduction. And of course, you can upregulate that gene and you get a heck of a lot more paracamarate. So that defines that you were right about the hypothesis that there is a gene and enzyme that's involved in making those conjugates, and that's how these things get into lignin. They don't occur at the polymer stage, they get on the monomers. Okay. Oh, sorry. And just today, some of you have heard probably there's CRISPR methods now that are really powerful for knocking out genes. So here's a corn lignin with tons of paracamarate in it. Just by the way, again, you all know this, but it often escapes people's notice that NMR is so much more diagnostic because of the number of constraints that are on any structure you think. If you have a peak that's here, it's already pretty cool because it's got a carbon shift and a proton shift. But if these other peaks aren't here, then it's not paracamarate, right? It's diagnostic as heck. Only if you see these other components as well. All right, you knock this thing out and they're gone. Okay, but again, there's nothing much more clear than looking at the NMR to see what you've done here. It's as clear as day, and any other technique, it's very difficult. And you get a nice paper out of it. Great. Right, next I want to get on to the power of 3D NMR and models for assigning peaks in a complex spectrum, and illustrate this with another one of these transferases, but one that just puts acetate on monolignals. Quite honestly, we've been looking with Sean Mansfield's group for this for quite a long time, and Douglas at our lab successfully made poplar transgenics already, and I want to show you how nicely NMR allows us to prove that we've done what we were intending to do by finding that gene and popping it into poplar. So a bit of a case for 2D, 3D NMR and for model compounds. All right, so here's the side chain region, these same A, B, and C units that I was talking about before in the lignin polymer. Here's what the transgenic that Namdagila made looks like. And if you're used to doing those YouTube spot the difference tests, you'll note that there are some things that are different between these. And you can start to go ahead and assign them and label them. But how do you do that? How do you prove, first of all, how do you know what they are? How do you prove that those were really from acetates? And how do you make authentic assignments of those things? So, first way, love 3D NMR. Complex as heck. 3D, yeah, right. The F2, F3 planes are proton carbon spectra, so these are HSQCs, but at each proton shift. So in principle, if a proton is separated from other protons, you can get an HSQC of just the one component on its own plane. So, for example, we look at this proton shift here where the B component is. Sorry, there's a bit of noise in it, but there's some other peaks that correlate in there. And so we're assuming this is alpha, beta, and gamma in the same structure. You slide it across and you realize you've just explained, yes, alpha, of course, but also this green peak must have been assigned right. It seems to be gamma, and this green peak is beta for that structure. So very powerful when you can isolate a component on its own plane. You move over just a tiny little bit and suddenly two other peaks start showing up. I can't see it on my screen. Sorry, there's another peak up here that are in red. You slide those across, and I've just assigned these new peaks, and this one's still in the same place. So in other words, what we're saying, and sorry, I'm assigning these to be the acetates. We'll prove it again in a second. Assigning these to be the acetates, the new structures that we've made in this engineered plant. The betas moved up to here when you acetylate it. The gammas moved down to here when you acetylate it, and this thing didn't move. Cool. The way to really prove these things is to also have model compounds. So we make a model compound for a guaiacyl unit, 1-methoxyl. We've esterified it here. We make this model and again slide them over, and you see you've identified these green peaks, beta, gamma, alpha. Fine, fine, fine. What am I doing? You acetylate that, slide those over, and you realize, ooh, I've just basically confirmed, assigned these new peaks. Again, beta went up to here when you acetylated it, gamma went down to here when you acetylated it, and this guy didn't move. Yeah? And there was a peak hiding under some protein peaks that are really annoying. This is this N group peak that slid out all by itself when it got acetylated as well. So an extra set of peaks that we can assign. Just very quickly, beta ethers, same kind of thing. You make the normal model. Okay, so the guaiacyl beta ethers here, the beta correlation is here for guaiacyl, gammas is here. Wonderful. You take a syringal one, slide that over. Syringals are to the left. The syringals are down here in the beta. These are about the same. You make the acetates and you slide those over, and you realize, oh, all these new peaks down here have something to do with gamma. This guy slid way up to here, out of here from up to here. This guy didn't move. So basically, again, the gammas went from here down to here. The betas went from here up to here, and alpha hardly moved. Last one, syringal, yeah, big deal. Syringal, here they are, still over here. The syringal gammas, beautiful again, explaining these purple peaks. The syringal peak went from here into here, so it's going to be overlapped by guaiacyl peaks, and we'll never be able to dig it out of there in the complex spectrum. But at least we sort of know where it must be showing up now. Yeah? Right, same thing. Gammas slid down, beta went up, etc. Okay, so in those spectra, what you've done is basically assign now all these differential peaks. We've got the green set, the new green set that wasn't in the original. We've got the new blue set that wasn't in the original. And we got this thing on the end that also wasn't in the original. So basically, you've assigned absolutely everything that's differential in that spectrum from the transgenic that has that acetate gene put into it. So we've made pretty dang sure that we've got these gamma acetylated lignins, and we've made gamma acetylated monolignals to make that lignin. Yeah, I'll try and do this quickly because you'll know this. Of course, there are other planes in a 2D. There's a F1, F3 plane, which is proton-proton at given carbons. And this is useful because the carbons are often better dispersed. So just quickly, for example, here's that C structure. If you look at the correlation at this carbon, you see a lovely little toxy spectrum, all the protons in that coupling network. If you look from the beta carbon, you see the same set of protons in that same coupling network. Brilliant. You can do that for beta ethers, and I won't go into it, but basically you can separate on their own planes the trio and erythro isomers of the syringal compounds, the trio and erythro isomers of the guaiacyl compounds, all on their own plane and all just clean as heck. Unbelievable. And same for the acetates. All right. So, okay, some of you are asking, well, who cares about isomers and S to G? Believe it or not, that's really important. Process engineers care. And the reason they care is when you're trying to delignify, you're breaking up these things, and the isomers behave differently. The erythro cleaves faster than the trio isomer, and the syringal cleaves faster than the guaiacyl isomer. And by the way, when you have two components that cleave at different rates, the mixture of those two components is not some average of that, of course. It's not even linear. And so that became a recognition for a while. But basically, that data is important, again, for modeling things like delignification, pulping, for example. Okay. Last thing. We've been lucky enough to have collaborators that have been making C13 enriched biomass. We love the 10, 12, 15% enriched biomass because you can do all the same experiments as you can do with unlabeled materials without any changes, and you just get a great sensitivity enhancement. But we've been getting these 100% labeled materials recently, and these are both great and a pain in the butt. First of all, so here's a normal old HSQC of 100% labeled sorghum. It happens to be a grass lignin again. And I want to just concentrate at the moment on these T peaks, these tricin peaks. I'll explain those in a second. Clearly, there's some multiplicity here that is not in the normal spectrum. Usually these are just single peaks, but we're getting some effect due to the carbon-carbon coupling. This isn't actually the carbon-carbon coupling constant, by the way. There's some residual effects from the experiment. But anyway, you get a dispersion into a big mess of these peaks that make for not clean experiments. How do you deal with that? There's constant time experiments that help collapse these down to single peaks again, like in a normal HSQC. The only issue with the constant time experiments is that that time period is crucial and dictates the intensity of the various peaks. So they actually vary with that time period. So we find we run two experiments, an 8 millisecond, a 14 millisecond one. This tricin peak, for example, shows up just magnificently in the 14 millisecond. It's less here, but some of the other peaks are more intense. So the quantitative aspects are sort of gone a bit, but you can always find all your components basically by running a couple of experiments. Again, these tricin peaks are collapsed very, very nicely when you do those constant time experiments. So that gets you back to realizing some of the sensitivity enhancement that you expected to gain from having 100% labeled material, 100% greater in principle signal to noise that isn't realized if you don't do these, if you do the normal experiments. All right, and I'm sure all of you knew that as well. All right, Jorge identified these tricin components in lignin, and I just have to mention that because this was an amazing discovery at first. Lignin was thought to be made from the monolignols. This is a flavonoid from totally outside the pathway and turns out to be incorporated into into grass lignins and other lignins. Turns out to be very, very valuable, and we didn't even know it was sitting there to be capitalized upon until these lovely NMR experiments. Marco Tonelli at NMR FAM and Clemens Anklin at Brooker really helped steer us onto these CT experiments, and these wonderful people made the C13 labeled sorghum for this work. One more experiment. Because you have full carbon labeling, so the 10% labeling is great for getting HSQCs, HMBCs, and you can see we're getting some nice correlations. They're horrible peaks again because of the horribleness of our samples. But you can do these carbon-carbon correlation experiments, the same kinds as the COSY or TOXY type experiments. And let me highlight that. We'll look at these tricin peaks again. So the very next slide, I've colored the tricin peaks, and look at all the correlations for tricin peaks. All of these red peaks all come from tricin. Some of them, of course, are quaternary positions, carbons. Some of them are protonated that you wouldn't have seen before by your other experiments, all just made possible by doing these carbon-carbon. It turns out to be a carbon-carbon FLOPSY experiment. I'll comment on that in a second. These paracamarate peaks, they show up. Look at all these correlations. Unbelievable. Unbelievable. And then the cis paracamarate that we're also seeing now. Whoops, that didn't show up yet. Where did it go? Here we go. These blue ones, a bunch of these are showing up as well. So cis paracamarate formed by photochemical reactions in the polymer. Once again, Guy Liffens in Toulouse created, if you like, but produced the experiment, well, modified the experiment for this particular C-FLOPSY that we used for that wasn't relevant for the protein work, but is very nice for our kinds of samples. And just love that experiment. It's got unlimited potential. And again, Jorge Rencoret realized what these blue peaks were all about. Cis paracamarate. He's got a nice paper on that recently. My final point is, how the heck would we be here? How would we have all this knowledge about the structure and stereochemistry and everything else without NMR? Can't imagine. It is so diagnostic, so so impressive at what it can tell you, and I hope you have a little more appreciation for both for lignin itself and for how NMR helps with even a horrible polymer. And by the way, I forgot to mention the whole cell wall. Whole wood, for example, solution state spectra by those experiments almost look identical in some regions. Obviously, they're overwhelmed by polysaccharides in other regions, but amazingly useful. All right, thanks very much.
Okay, do we have anybody with
Oh,
questions?
yeah. I'll also jump up here and ask the same question. I'll have some in a minute. I love how enthusiastic you are about seeing correlations and seeing what you can see. I mean, that excites me too. But one question more broadly, what I see that you have a whole group of collaborators all working on lignins, and what is the societal goal ultimately that's driving all of this?
Thanks. Basically, a couple of things. One, it is always just been a waste product. It's been burnt. You isolate cellulose by pulping or whatever, and you basically burn, throw away. There's value to burning, by the way. It drives the process a bit. But when it's realized that that's the largest sustainable source of aromatics on the planet, and we're not utilizing it, it's almost tragic. It's really potentially incredibly useful. It's been underutilized, and that's suddenly being recognized in the biofuels and other industries. So we're making Tylenol from it and all kinds of things these days. Of course, Tylenol's a bad word today, sorry. Anyway, yeah, there's a heck of a lot you can do with the polymer once you realize what it is and how to not wreck it when you're doing things like pulping or isolating your cellulose. Usually we don't think about it up front. We've just been trying to get rid of the dang thing away from the cellulose. Now we think about trying to preserve it up front so that we can do good things with it, and get value from it.
So is the mix half and half specialty chemistry and versus fuels?
Indeed, and polymer chemistry is still there again. Often you take it back to the monomer and polymerize something again. We've got sort of BHP, what are those things called? Oh, for goodness sakes, bisphenol A equivalents from lignin now that are less estrogenic. There are polymers you can make from it. As a polymer itself, it's actually quite useful, quite hard to deal with because it's the structurally very complex. So, but you can make nice carbon nanotubes and so on apparently with these things. So, yeah, there's a bunch of uses. Anyway, there's been a resurgence in this. An interest in this. It was always considered too difficult a dang polymer. I mean, it's a stereochemical nightmare. I didn't quite say it, but you only need a 20mer and you have tens, hundreds of billions of isomers. There's no two molecules the same. So it's a shocker.
Oh, great. So I'll go on, and I see John Court has his hand up.
Hi, John. I was wondering if you had attempted 3D or even 4D HCCH toxi or cozy type experiments. So spread your FLOPSY out into one or two additional proton dimensions.
I'm gonna say no, I think. So the HSQC Toxi 2D that we run on 10% labeled and so on, that works incredibly well. Yes. And that's I sort of used that on that dimer mix a bit. For the fully labeled materials, I suspect I need to do more research to figure out how the heck we can implement some of those. So
Yeah, they're used for proteins all the time.
they
And
are. Every
yeah,
time we've looked at a protein sequence, though, you always have the carbonyl shift in there and all kinds of things, and you're just going, oh, but we don't have any of those. So anyway, they require a bit of tweaking, and that's
Actually,
where
John,
Guy
so you,
Lippens really helped
yeah,
us enormously with the
John,
H
I'm
linkers.
here. So, and I
Oh,
actually ran them yesterday.
oh,
I ran them yesterday, and even better, I solved the HMBC problem on the
oh,
C13 100%.
what?
Yeah,
Okay, because we failed spectacularly with HMBC. I know some people have some equivalents for fully labeled materials. We couldn't get them to work. You've done it,
I
Guy?
get your trisim perfectly out,
Oh
the
my
full
God.
chain. Yeah.
Okay, talk to me. So we're lucky
So
to have connected with Guy Lippens in France. Oh my gosh.
So I see Futhi Fong.
Yes, I have a question because I noticed you can detect amino acid in the biomass. I think it's phenylalanine and tyrosine, I guess. How you know it's amino acid and how could we remove it from the lignin or from the biomass?
You're very astute. These are a pain in the butt for us. We don't want them. They shouldn't be part of the cell wall, but they do get trapped in the wall, and they might even be part of the wall in some ways. You can remove them by doing pronase treatment, for example. That works quite well. Hoon Kim has a lovely paper on that to clean up the lignin. We find it's not usually worth it. You can get most of them out by simple extraction, actually. So you do water extractions up front on the finely divided material and get rid of them first. But yeah, they're in there. You're right, and yeah, mostly they're annoying to us.
Yes, it's annoying in my research as well. I'm trying to find a way to remove it.
Okay. So pronase works really well. So a paper by Hoon Kim. Um,
Thank you.
if you send me your email, I can send you that.
Yep, thank you very much.
So somebody else speak up with a question. Well, I've really more questions. I'll ask again. Okay. Well, I've really enjoyed this talk and the enthusiasm and this and all of what you learned.
There's some messages in the chat
Dan,
room, Dave. There's some
what?
messages in the chat.
Oh, okay, great. Usually I'm not the person that is I'm not used to it here.
Oh,
Yeah.
yeah. Do you want to read them, David, or
Yeah,
I
I
can
will.
see some here. Okay.
Usually we have two people, so there's always one other person looking at the chat. And
Could you,
okay, so let me go
best
from
would
the
be
top.
to ask the best would be to ask the people to actually
Yes,
ask the question rather
right.
than
Okay.
read it out.
So I see the first chat is from Erica Brendler. Is she here? Well, the question
Yes,
is,
yes, I am. It's no
okay.
question. It was just a comment because you asked what you can do with lignins. So I know that you can produce some chemicals from it, or that it's for some groups, it's the attempt to find out which chemicals you can produce from this product. So vanillin is one of them.
It certainly is. It was one of the very first ones that was ever produced. In fact, the world supply of artificial vanillin is from lignosulfonates, but sulfite pulping. Yep, that's where the lignin is. Where the vanillin is produced from. In fact, it's not a great yield. Funnily enough, you have troubles with getting vanillic acid and all kinds of other things with it. And if you do hardwoods, you've got the syringeal analog as well. So a bit of a pain. But yes, that absolutely is commercial.
So there's another question in the chat from Rashed. Is he here to speak up? Or she?
Oh yeah, I was just because I've just started learning about lignin. I just wanted to see if there's a database for the NMR shifts, and some people kindly replied with some citations, which was quite nice.
Indeed.
Oh, okay.
Thank you.
Can send you plenty more. We actually have a database of model compounds as well that's out on the web and freely available. But yeah, this little mix of the three compounds, I almost suggest making that little mix because then you can you can assign directly from your model spectra and your polymer spectrum, you can assign straight from those and make sure you haven't assigned anything wrong by looking at papers. It's a pretty good way, and it's very easy to make that mix. Hopefully we'll have a paper out on that fairly soon. The trouble is it's a little bit simple that we're not quite sure where to submit that. It's useful, but it's it's quite simple.
So I see that Jorge put in a link for the database.
Oh, indeed. Apparently so.
Okay, and also I'll just repeat, unless Salvador, if you'd like to speak up, he's also suggested a paper.
Yep.
Structural.
There's a question on 3D NMR pulse sequence by Alyssa.
Okay, just a second here.
Well, Salvador, just to answer yours, you're right. Jorge Rencoret, freaking awesome, and he's in Jose Carlos's group in Spain. They're amazing. They have some beautiful papers with lovely assignments. Yuki Tobimatsu in Japan, and I suppose some of our own, and many others. But there's lots of papers with assignments, and yeah.
I see a question from Alyssa about the 3D sequence.
Yep. Yes, it is
Alyssa?
part of the. Yes, it is standard.
Yeah. Okay. Great.
Yep, it's MLive 3D something or other. I can send you that, Alyssa. I think I know who you are. Yes.
Okay. And Jorge's comment about Beauregard, is that a company or?
It is.
And then there's a link. Okay.
Yep.
And
so there used to be this lovely comment that you can make anything from lignin except money. Don't tell that to don't tell that to Beauregard. They're doing extremely well.
right. And
That's a question by Darcy.
Yes, right. Darcy?
Darcy. Ah.
Can you hear me?
Yes.
Yep.
I just was curious about the 3D experiments as well. I guess tacking on from Alyssa's question, how long are you typically spending to acquire that data? With the caveat being, I'm sure there's sample dependence and labeling dependence on that answer.
Yeah. So it was a bit of a same issue that we used to have when 2D NMR first came out. We were used to running carbons and protons, and we know how long carbons take, and we always thought that that 2D, which you acquired as carbon spectra in those days, by the way, you thought it had to take way longer than a 1D experiment. This is not true. The total number of scans over your entire experiment dictates the signal to noise in that experiment. So a 3D doesn't necessarily take any longer than a 2D. You often want enough data points to in all the dimensions to have the resolution you want. So you need quite a few actually. So we typically take 24 to 30 hours to run those. But that can be on unlabeled material as well. Same things. The sensitivity is actually plenty there. It's the number of cycles you have to do through the various dimensions that dictates the time in the end. Just backing up again too, you think, 2Ds, everyone runs them for a very long time. The whole cell wall NMR spectra that we run, you can run a very good spectra in 5 minutes. 5 minutes. You couldn't have imagined that way back, but with the quantum mechanical trickery that goes on with making the HSQC experiments, acquiring the proton, doing the first pulse on proton, the sensitivity enhancement there is a factor of 30 or something over the old experiments. It's just remarkable.
Yeah, that's awesome. Thanks. I guess an add-on to that, if nobody minds, but it'll be quick. Does NUS work with this?
Sorry?
Does non-uniform sampling work with this?
Yeah, absolutely.
Yeah, I see that in the chat.
We're being a bit remiss there, actually. It's a very good thing to do. We have a luxury, I guess, of having two machines and enough time on them that we haven't always gone there. And I'm a little bit embarrassed about that. But yes, other people are big fans of NUS. They absolutely do. Only gotcha to pay attention to is if you have really rapidly relaxing samples like our ones, these whole cell wall samples that you can turn the tube upside down and it doesn't go anywhere. They're horrible. They relax in about 30 milliseconds or something or other. with those samples, you better front load your NUS. If you have it in the normal, going out for a second or something or other and have your data points that you're sampling way out beyond where there's any signal, you're wasting your time. So there's two kinds of NUSs. One is front loaded, and just look into that if you've got these horrible samples. But otherwise, yes, absolutely.
Thank you.
Okay. And Eric asked what percent NUS you use, but I think you answered that.
Yeah, 50 or 25 for us, but yeah, we don't do enough of it. We're very bad.
Nice having your own machines.
Isn't it nice? And it's lovely when Obama gives it to you. It's just wonderful.
Are there more questions? We've run out to the end of the chat here. Are there any more questions from people? Well, okay. Well, if there are no more questions, I'd like to thank John for a really great talk, and I've been excited by it, and I think others have. And so with that, if there are no more comments, I'll give it back to John if he's still here.
I think he's left us for today, but I just will again ask questions of people if they have any knowledge of anybody that wants to present. Contact us through the Ivan MR website, ivanmr.com, and we can try and get you on the schedule for 2026.
Yeah, I guess I would say Eric is taking a very strong role in finding speakers for us. I think Chris used to do that a lot, but Eric is doing that a lot now. So get in contact with him if you want to speak. If there are no more questions or comments, I will conclude our meeting. So thank you, everyone.
Thanks very much, guys.